few bacterial diseases. Among them, we have seen typhoid disease, which is caused by Salmonella typhi. Then we have seen cholera disease, which is caused by Vibrio cholerae bacteria. Later on, in second unit, we have seen two diseases caused by two different bacteria. In that, the most important disease which we have studied is the pulmonary tuberculosis, which is caused by Mycobacterium tuberculosis. Then, in that unit, there is another disease is also there. That disease is also a bacterial disease, and that disease is known as a syphilis disease, and this is also a bacterial disease which is caused by a group of bacteria which belongs to a genus Treponema. Now this group of bacteria have characteristics that these bacteria, they are elongated, motile, flexible bacteria which are twisted spirally along the long axis, and such group of bacteria which shows these characters, such group of bacteria, they are known as spirochetes. The name spirochetes, it is derived from a Greek word spira, means coil, and chaets means hairs. So this is the group of bacteria which is involved in causing the disease syphilis. Now structurally, when we observe these bacteria, they are more complex than the other bacteria, like that of cholera bacteria, typhoid bacteria, or that Mycobacterium tuberculosis bacteria. This group of bacteria, they are more complex than these bacteria, which we have seen in earlier classes. The characteristic of this bacteria is the presence of varying number of endoflagella. This group of bacteria shows a character that in this there is a presence of endoflagella, which are polar flagella coiled along the helical protoplasmic cylinder and situated between the outer membrane and the cell wall. There is a presence of endoflagella. Means these flagella, they are present inside the bacterial cell, so that's why they are called as endoflagella. This is the characteristic which is shown by this Treponema bacteria. And these endoflagella, they are believed to be responsible for the motility of this group of bacteria. But the exact mechanism, how this bacteria moves, or how the movement of this bacteria takes place, is not understood. And these spirochete bacteria, they are also showing variation in their size. Some of these bacteria may as long as 500 micrometer long. Some of them, they are as much as short 5 micrometer. And so there is a variation in the size of this group of bacteria. Some of them are large, having size up to 500 micrometer. Some of them are very, very short, having size of 5 micrometer. And these bacteria, when we stain these bacteria by using gram staining, they are gram-negative bacteria. And most of this group of bacteria, they are free living saprophytes. Means they can grow on dead and decaying organic matter. But some of them, they are obligate parasites. They are obligate parasites, means they grow only in the living host, like human beings. And when we observe the respiratory character of this group of bacteria, they are aerobic, anaerobic, and facultative nature. Means this group of bacteria, they can survive in the presence of oxygen, in the absence of oxygen, or in both the conditions, in presence, in the absence of oxygen. That is the facultative condition. And this group of bacteria, they replicate by a mechanism. That mechanism is known as a transverse fission. By this mechanism, it multiplies, and the multiplication time of this group of bacteria is very, very long, as like that of pulmonary tuberculosis bacteria. Now, this group of bacteria, these are the characteristics shown by this treponema. And this word treponema, it is derived from the Greek letter tripos, means turn, and nema means thread. So when we look this bacteria under the microscope, it looks like the threads which are having turns in it. You can see here. This is the microscopic observation of this treponema bacteria. You can look here. This look like a threads, but having turns in it. So that's why the name treponema, it is derived there. And these treponemas, they are relatively short, slender spirochetes with fine spirals and pointed or rounded at the end. You can see these are spirals, and they at the end, when we see the ends of this bacteria, they are either pointed end or rounded ends. Just a second. And some of the members of this treponema group, they are pathogenic, and some of them, they are also commensals. They are most commonly observed. This group of bacteria, those which are commensals, they are commensals in mouth, intestines, genitalia, and these are the particular locations which this group of bacteria, they are most commonly found as a commensals. Now, some of the members of this group of bacteria, they are pathogenic, and these are not only pathogenic, but they are obligate parasites. Means we cannot grow such a bacteria in artificial medium. Which we use to produce in our laboratories, such as nutrient agar, McConkey's agar. On such mediums, these bacteria, these obligate parasitic bacteria, they are not able to grow. Here in this case, we require them to grow on living cells, like that of tissue culture cells. On these cell lines, they can easily grow because they are obligate parasites. They are not grown on artificial media where there is a cell-free medium. Means there is no any kind of living cells are there. So in such a medium, these bacteria are not grown. But some of the commensals, they may be grown on artificial media. But the obligate parasitic bacteria of this group, they won't grow. They won't grow on artificial media. They require cells to grow. So that's why they are obligate. Means compulsory. There is a requirement of living cell for this group of bacteria to grow. And one of the member of this group of bacteria, which is responsible for causing a disease, that disease is known as a syphilis disease. And the causative agent of syphilis disease is the Treponema pallidum. This is the bacteria which is responsible for causing the syphilis disease. Now in this disease, firstly we have to see the etiological characteristic of this disease. Means their cultural, morphological and biochemical characteristic of this pathogen, which have to see in the etiology. This Treponema pallidum bacteria, which is the causative agent of syphilis, was firstly discovered by the scientists Shordin and Hoffman in the cankers and inguinal lymph nodes of syphilitic patients. They firstly observed this bacteria in the cankers. That is a character which is shown by this disease. Where this bacteria grow, it produces cankers. In that canker, and some of the lymph nodes, ingenial lymph nodes, this bacteria, they have been observed by these scientists. And the name pallidum, it is given to this group bacteria because it stains pale on staining. When we stain these bacteria, they are not taking the dark color. They show pale color, light color. So that's why the name pallidum is given to this bacteria, Treponema pallidum, which is the causative agent of syphilis disease. Now when we see the morphological characteristic of this bacteria, they are thin, means they are thin in size, delicate spirochetes with tapering ends, means at the end there is a pointed ends are there, and which is having size about 10 micrometer long and 0.1 to 0.2 micrometer wide. Means this bacterial size, there is a variation in the size of this group of bacteria. This bacteria Treponema pallidum. Now this bacteria have about 10 irregular spirals. When we observe the morphology of this bacteria under the microscope, you are able to observe there 10 irregular spirals are there, which are sharp and angular, and at a regular interval of about 1 micrometer. 1 micrometer, there is a spiral. Means there are at every 1 micrometer length there is a presence of a spiral. Spiral is there, and this bacteria, it is actively motile. This motility is shown like that of when we use the screw and tighten it with the help of screwdriver. Then when we move the screwdriver, the screw goes inside. Like that, this bacteria shows that type of movement. And this is because of the presence of these endoflagellas in the bacterial cell. And these spirals, this spiral shows the presence of endoflagella. And this endoflagella, as it is moved, you can see here, this is the structure of this Treponema pallidum. These are the spirals, and these are nothing but the endoflagellas. And this bacteria moves like a screws, backward, forward, and like this bacteria shows the motility. So that's why this bacteria, it is a motile one, and this motility, it is because of the presence of endoflagellas in it. And because of this character, they show the mobility. And another important character which helps this motility, it is the flexibility of this bacteria. These bacteria, they are flexible, not hard one. So that's why due to the movement of this spiral movement, it is caused by these endoflagellas, and this is possible because of the flexibility which is shown by this bacterial morphology. This is the structure. These are the endoflagellas. This pink color, these are the endoflagellas. This is the axial filament, cell membrane, cell wall, plasmic space, and outer membrane. Same is the figure also. You can see the tapering ends which are present at the end of this bacterial cell. This is the figure. This is the photograph after the staining. You can see here this bacteria, it doesn't take the dark color, it takes the pale color. Because of that, it is known as a pallidum, Treponema pallidum. And this bacteria, it is a gram negative bacteria. Means it takes, when we stain this bacteria with the help of gram staining, it takes the pink color, and which is of light in nature. Then this bacteria cannot be seen under the light microscope in the wet films. Means in the hanging drop technique, we are not able to visible this. We are not able to see this bacteria in the wet films under the light microscope. but it can be made visible by negative staining with Indian inks. So when we use this negative staining, this negative staining stains the background of this bacteria. So we are clearly able to observe the morphology of this bacteria. And the morphology and motility of this bacteria can be easily seen under the dark ground or phase contrast microscope. When we use this microscope, then we are able to cleanly observe this Treponema pallidum bacteria. Now this bacteria, it doesn't take ordinarily bacterial stain, but it stains light rose red with prolonged giemsa staining. When we try to stain this bacteria, it doesn't stain ordinarily with the bacterial stain, but when we use giemsa stain, it stains light rose red, and it can be stained by using a silver impregnation method, and through this you can observe this bacteria. Now there are various methods are available for staining this bacteria. Fontana's method is useful for staining films. Levedecky's method used for staining tissues sections where these bacteria are present. So this bacteria can be observed either by using negative staining, or we can observe this bacteria under the phase contrast microscope or dark ground microscope, or we can stain this bacteria by using giemsa stains. We can observe this bacteria by using silver impregnation techniques, or we can observe this bacteria in the films by using Fontana's method, and by using Levedecky's method we can observe this bacteria in tissue sections of that patient's different organs. Now next character is the cultivation character of this bacteria. These pathogenic treponemas, they don't grow in artificial medium. The limited growth of Treponema pallidum has been obtained when we grow this bacteria, or when we cultivate this bacteria in tissue culture cells. And it is possible to maintain this Treponema pallidum bacteria in motile and virulent form for near about 10 to 12 days in complex medium under anaerobic condition. So ordinarily this bacteria is not able to grow on artificial medium, but in order to grow this bacteria in laboratory we need the co-cultivation with tissue cultures. This is the condition. Tissue culture growing this bacteria in a co-cultivation manner in tissue culture. This is the condition in order to grow this Treponema pallidum bacteria in the laboratories. But it can be grown on a complex medium under the anaerobic condition, which is maintained 10 to 12 days. A virulent Treponema pallidum strain, it can be maintained for many days or many decades by serial testicular passage in rabbits. So in order to cultivate this bacteria and maintain this bacteria, we can use living animals such as rabbit. In rabbits, we are going to use the testes of this rabbit where this bacteria we are going to multiply, and we can maintain this for longer period of time or longer decades. And one such strain, Nicole strain, it is isolated from the brain of fatal case of general paralysis of the insane in 1912, is still being propagated and used for diagnostic and research purposes. Among the different strains which are present in Treponema pallidum, one such strain, that is Nicole strain, it is isolated from a patient which is having a general paralysis, and this strain, it is continuously grown by using this serial testicular passage in rabbits. So this strain, Nicole strain, it is maintained live condition in the testes of the rabbit, and we can multiply them by inoculating this culture into the testes of rabbits. And this multiplication or this cultivation of this bacteria in this living organism, it helps to us to study them. As you know that this bacteria is not able to grow on an artificial medium. So we have to use the living cells in order to grow this bacteria. And by growing this bacteria, we can use these bacteria which are grown on these living cells for diagnostic, development of diagnosis, or other research purpose, development of vaccination, development of treatment process. In such cases, we can use this grown pathogen. You can see here the rabbit, it can be used here to grow this bacteria and multiply this bacteria in rabbit as a living. So this bacteria can continuously multiply in the testes of these rabbits. Then the resistance character of this bacteria. This Treponema pallidum bacteria, it is very delicate and it is being readily inactivated by drying or by heat. So when we heat this bacteria or when we keep this environment dry, then this bacteria, it easily get killed. When we heat any medium or any object up to 41 to 42 degrees Celsius temperature for 1 hour, this bacteria, it is totally destroyed then. Hence, the fomites, they are of little importance in the transmission of infection of this syphilis disease. This disease is not transferred by fomites, means in inanimate objects. They are not act as a vehicle for the transfer of this infection into healthy persons. Now, the susceptibility of the Treponema pallidum to heat was the basis of fewer therapy for syphilis disease. As you know that during the treatment, various treatment procedures are available. In that, fewer therapy, where we enhance the temperature of that person which is having this infection. So that's because of this pathogen can be easily destroyed. This pathogen can be easily killed in 1 to 3 days at 0 to 4 degrees Celsius temperature. So the transfusion syphilis, it can be prevented by storing the blood for at least 4 days in refrigerator before transfusion. So we have to avoid the transfusion syphilis, means which is transferred because of the contaminated blood samples. That blood sample which we have collected during the donation process, they should be kept in the refrigerator for 4 days before transfusion process. So all the blood, it is kept in the refrigerators, cooling the refrigerators. So because when we keep this blood in these refrigerators, the temperature of this refrigerator, it is up to 0 to 4 degrees Celsius temperature. And then in this temperature, this bacteria is easily killed. And stored frozen at minus 70 degrees Celsius temperature in 10% glycerol or in liquid nitrogen. When we store this bacteria in frozen condition at minus 70 degrees Celsius temperature in 10% glycerol or in liquid nitrogen, this bacteria can remain viable for 10 to 15 years. So in this manner, we can keep the bacteria as a sample in the laboratories by freezing them in this 10% glycerol and keeping them at minus 70 degrees Celsius temperature. or we can frozen them in liquid nitrogen and kept in the refrigerator so that bacteria can remain alive for up to 10 to 15 years. Now this bacteria, it is sensitive to some of the other components which we use in our day-to-day life. This bacteria, it is inactivated by contact with oxygen, distilled water, soaps, arsenals, mercurials, bismuth, common antiseptic agents and antibiotics. Means this bacteria, it is sensitive to different components which we use in our day-to-day life, such as soaps, common disinfectants. These are the helpful methods which prevents the growth of this bacteria. So this bacteria can be easily destroyed by using these antiseptics, soaps. Then the next point in this is the epidemiology. Up to now we have seen the etiological characteristic. In that we have seen the morphology, then cultivation, then resistance characteristics which are shown by this Treponema pallidum bacteria, then the epidemiological characteristics. Epidemiological characteristic, it includes how this infection, how this disease is spread among the community. That we call it as a epidemiology. Now venereal syphilis means those syphilis which are transferred due to the sexual contacts. That disease we call venereal syphilis. Means such syphilis disease which is transferred due to the sexual contacts. Such syphilis disease is known as a venereal syphilis, and this is worldwide in distribution. Now during the 5th century, it has been recorded and studied. After the discovery of this pathogen, we have studied and recorded this bacteria. This disease has undergone much variation in its natural history and clinical features. So this venereal syphilis, which is caused because of the sexual contacts between the healthy and patient, during this there is a transfer of this infection from healthy person to the patient to the healthy person. And when we recorded the conditions and the history of this disease, it has been shown that there is a variation in its natural history and clinical features. Originally it was described as a highly virulent disease with florid cutaneous manifestation. Earlier this disease was very highly virulent, means highly spreading, which is mostly causing the cutaneous manifestation, showing the cutaneous symptoms on that persons. With the discovery of dramatic therapeutic response to penicillin, this penicillin antibiotic, it is useful to the treatment of the patients with this syphilis disease. This bacteria can be easily killed by using the penicillin. It was hoped that with the help of penicillin we can eradicate the syphilis disease, because this pathogen is not affecting any other host except the human beings. The only source for this pathogenic bacteria is the human beings only. There is no reservoir host is there. So it is hoped that by the discovery of the penicillin, this disease, syphilis disease, will be eradicated. But such elimination of disease hasn't been possible because of the various factors changing the customs, habitats and values in society. These are the responsible factors because of which this syphilis disease wasn't being eradicated with the help of penicillin antibiotic. So it remained there. Now with the development of AIDS pandemic, which has had an impact on syphilis. As you know that AIDS, it is also a sexually transmitted disease like that of syphilis, and in the most places the fear of AIDS and safer sex practices leads to the fall in the incidence of syphilis and all the sexually transmitted diseases initially, but this trend didn't continue everywhere. In some countries these precautions they have been taken, but in some of the countries these precautions they hasn't been taken there. So that's why this disease remain there, because the only source of this pathogen is the human beings. If this disease is eradicated from human beings, it will not affect any other organism, any other animal, because the only source on which this pathogen can affect is the human beings. Now the co-current infection with syphilis and HIV is the common one. Most of the HIV infected persons or AIDS having patients, they have co-current infection of syphilis in them. This is the most common one and may lead to the earlier evolution of neurosyphilis. In such patients which are having AIDS, there is a development of another syphilitic condition, that is the neurosyphilis condition, which is developed among the patients which are having AIDS, and there is also an infection of syphilis. In such condition there is a development of neurosyphilis condition is there. Okay, so this is all about the epidemiology. In that, this disease is mainly transferred through the physical contacts, and that physical contact is the sexual contact. And when a infected patient make a contact with a healthy person, then this infection can be transferred to the healthy person. So the only mode of transmission is the contact. No other mode have been observed here in case of syphilis disease. Fomitis, it doesn't have any role in the transfer of syphilis disease among the healthy persons, but the only mode is the sexual contact. It is the only mode through which syphilis disease can be transferred from one person to the another person. So this is all about our